Well, welcome back again to the narrated presentations of our ongoing look at the chapters for this semester. Today we're looking at, or this week, we're looking at chapter 24, the origin of species. So just to remind you, in the previous chapter, we talked about how change can occur within a species, what is often referred to as microevolution. What we're looking at today is sometimes referred to as macroevolution. Now, one of the issues that most evolutionary biologists have with those two terms is that there's really no distinction between change within a species versus change that leads to new species. It's all the accumulation of adaptations through natural selection or descent with modification. The degree of change or the net effect of that change is what is reflected by the two terms. And it's sort of a way of deflecting some discomfort that some people have with the whole notion that species or new species can emerge as a consequence of natural selection. Most people don't argue that natural selection occurs, that change occurs within a species. It's very difficult to argue against that because it's readily observable. But observing the emergence of new species has been somewhat of a challenge. As I've indicated in the course of both of the narrated presentations and up to this point, there is evidence now that we can point to that demonstrates the emergence of new species as a consequence of natural selection in nature that can be observed in a relatively short period of time. And we will very quickly refer to that again, and I'm going to place the link to the videos that I've referred to previously, the videos from the grants, in the resources page, if I haven't already put it there, for this chapter, so that you can have an opportunity to go and take a look at them, because it really is some very unique and very important research that you ought to be aware of. So the first thing to remember is that descent with modification, natural selection, is a way of creating better adapted species or better adapted populations, because remember, the unit of evolution is a population. And we can explain very, very easily, and we can observe very, very easily, these changes within populations. We can see populations evolving over time. Not a big deal about that. But we also need to remember that the theory of evolution and natural selection also needs to explain the origin of new species. Because if we take a look at the fossil record, if we pay attention to what we see around the world, comparing existing species with what we see in the fossil record, we see very clear evidence of speciation, the appearance of new species. We need to bear in mind that over 99% of all species that have ever lived on the planet are dead, have gone, are extinct. And we only know of a few of them through the fossil record, and yet the fossil record shows us quite clearly that speciation is a reality, that new species do emerge. So we have to have a way within the framework of the theory of evolution and its engine, natural selection, to explain the origin of new species. And that can sometimes be very difficult, simply because it's very difficult to define what we mean by species. There have been many attempts to create definitions of species, and over time, about 11 or 12 different definitions of species have been generated because of the requirement to have a particular definition of a species in a particular situation. However, there is one species definition known as the biological species definition, which is the one that is largely accepted by the vast majority of biologists. And you'll notice that I'm being very circumspect there, and that's because there is still a significant amount of disagreement over what exactly a species represents. And how can you pin a label on a species by definition? But nonetheless, let's take a look at what the biological definition of a species actually says, because this is the definition that we're going to rely on throughout the course of this semester. be able to identify individual species. And so here it is. It's probably the safest definition of a species that you can put forward in a biological framework. And so it says that a biological species is defined as a population or group of populations whose members have the potential to interbreed in nature and produce viable, fertile offspring, but are unable to produce viable, fertile offspring with members of other populations. Now, there's a couple of key phrases in there that we need to pay attention to. So let me fire up my pen and identify for you those key phrases. So one is the potential to interbreed. Let's just eliminate that and put it in red so that it highlights a little bit better. So one is the potential to interbreed in nature. This cannot be something that is demonstrated in the laboratory. It's got to be in nature. Then they have to produce viable. That's the next part of it. And the next part is fertile offspring. And most people don't understand the distinction when we say viable and fertile offspring, when they first get introduced to this notion of a biological species. So what do we mean by these terms? Well, viable means that they survive to reproductive age. In other words, they don't die as embryos, they don't die in utero, they don't die in the eggs, they don't die as young individuals, they actually survive to reproductive age. That means that they then have the potential to leave behind offspring of their own. So that's what we mean by viable. They must survive to reproductive age or to sexual maturity, if you want to use another term. But that then also implies once they reach sexual maturity, the fertile part means that they must also be capable of actually leaving behind their own viable fertile offspring. As you'll see, we do need to apply both of those labels to the offspring of the individuals who have mated from the two separate populations. Not only do those individuals have to produce viable fertile offspring, but their offspring must be viable, or the offspring of the offspring must also be viable and fertile, because there is a reproductive barrier, and that's what this discussion is all about, reproductive barriers, that kicks in even at that stage, even if they are capable of producing fertile offspring. That's not a guarantee that these are still members of the same species. So once we get that definition out of the way, and you may already begin to see that there are issues with that definition, because it requires, number one, that the individuals be sexually reproducing. Asexual reproduction is not covered by this definition at all. It does imply also that the opportunity to mate must present itself within nature. It must mean that the individuals are observable. You can't apply this to species that are found in the fossil record. There's a whole number of a whole number of problems that go along with this species definition, because there's really not one single species definition that can be applied in all situations. There's no one size fits all definition, as you'll begin to understand as we continue on with this chapter. But before we go and take a look at what we can identify as the barriers that prevent these sorts of mating opportunities, what we should bear in mind is you can't just look and see that there are members of two different species or members of the same species. Physical appearance does not determine whether individuals are members of the same species or not. And these are two great examples. So the bottom one I'll talk about first, that's us, of course. There are a wide variety of morphs, physical appearances that we can identify in humans. Number of different populations on the basis of physical appearance. And it's not so long ago, in fact, maybe a couple of hundred years at the most, in some cases no more than 100 years, that physical appearance was actually used as a way of identifying individual quote unquote species within humans. And people were quite clear that there were the potential for a number of different species within Homo sapiens, and they were somewhat convinced of the fact. And in some situations that was used to discriminate between members of different population groups that were clearly physically different from the people doing the judging, which usually was Western Europeans. What we realize now is that no population of humans is reproductively isolated from other populations of humans. Now, there may have been in the past some tendency towards physical barriers that may have prevented easy breeding between human populations, but that didn't prevent it because humans are particularly mobile. And we see that reflected throughout the course of human history. And today it's very, very apparent, COVID notwithstanding, that it is easy for humans to move around because there is no place on the planet that you can't get to within 24 hours on airplanes. Yeah, I mean, it's that simple. And so the ability of humans to move and therefore to encounter individuals of different populations and potentially mate with them can no longer be considered to be a reproductive barrier. And in fact, the history of Homo sapiens includes mating with populations of related species, most notably the Neanderthals and the Denisovians that we are aware of, who have left behind genetic fingerprints in their own genome. So if you are someone whose ethnic origin lies outside of sub-Saharan Africa, you may have up to about 2.7% Neanderthal genes, particularly if you are European, Asian, Native American. There is a significant percentage of Neanderthal genes in your genome. If you're looking for Denisovian genetics or genes, you have to look in individuals who live in the South Pacific, the Melanesians and the Polynesians. Their ancestors migrating across Central Asia and Siberia encountered this second species of humans that we know were concurrent with or lived outside of Africa prior to Homo sapiens appearing on the scene. We only know them from a very, very limited amount of fossils. And in fact, the DNA comes from a small piece of finger bone. We don't have much more than a couple of pieces of finger bones to indicate that this group of humans existed, but we have enough to be able to do a complete genome sequence, and we know that their genome is different from Homo sapiens and different from Neanderthals. So they represent another species of humans that existed prior to Homo sapiens in Central Asia. That they're named after a cave in southern Siberia that existed prior to Homo sapiens migrating out of Africa some 100 to 120,000 years ago. We have actually pushed back in time the date where first a species appears in Africa, and second, when they began the migration out of Africa to give rise to Homo sapiens populations around the world. So we now think it's somewhere closer to 100 to 120,000 years ago, perhaps. But nonetheless, we know that these separate species did exist, but when we encountered them, clearly we could mate with them because their genome sequences are present in modern Homo sapien genomes. So no question that while we may have been reproductively isolated from them for some period of time, it wasn't sufficient, or from them via the common ancestors, it wasn't sufficient amount of time for us to diverge into separate species that were incapable of mating with each other once we encountered each other again. So that would suggest that it was speciation due to separation by distance, not separation by genetics. And we'll talk more about that as we dig deeper into this chapter. Then if you look at the picture at the top and you take a look at these birds, these are two males of the two species of the meadowlarks that we have here in the United States. The eastern meadowlark lives to the east of the Mississippi Valley. The western meadowlark lives to the west. The Mississippi Valley is a very common line of demarcation between eastern and western species here in the United States. But the Mississippi Valley also represents a range that is often shared by individuals from both species, and the ranges overlap. What keeps these two species isolated from each other, and yes, they are completely isolated from each other, they do not interbreed, is the fact that the two males sing different songs, and only the females of the appropriate species are attracted to that song. What caused these changes in song production, we don't know, but something changed in the past that separated these two populations that may have been a single species at one point, and they have diverged into two separate species, and they no longer interbreed with each other, yet they look almost identical. So physical appearance is not sufficient to determine whether individuals are part of the same species or not. We have to fall back on our definition, and we have to recognize that that definition implies within it this idea of reproductive isolation. Individuals that can interbreed, produce fertile and viable offspring, represent the same species. Those populations that can no longer interbreed and produce viable fertile offspring must be considered to be separate species. Speciation has either begun or has clearly happened, and that's why they are no longer interbreeding. So it's really all about reproductive isolation. And the question then becomes, what gives rise to reproductive isolation? And we recognize two basic types of barriers that prevent reproduction between populations, and we split them up on the basis of whether they occur before fertilization or after fertilization. So those that are pre-zygotic, these ones, occur prior to fertilization. Those that are post-zygotic occur after fertilization. So we have these two basic groups. And the textbook has a nice diagram that you can take a look at that presents these reproductive barriers as a timeline. So I have a slightly older version of the diagram, but nonetheless relevant, that outlines all of these. And I'm going to walk through all of these reproductive barriers in sequence to explain to you how they work. And particularly when we get towards the end, that's where a little bit of our misunderstanding often kicks in. And so I'll probably spend maybe a little bit longer explaining some of those than I would explaining some of the earlier ones. So the first one is kind of easy to understand. It's called habitat isolation. This is where species spend their time feeding and reproducing and living in two separate habitats. And as a consequence of that, individuals of the two species never encounter each other and therefore never mate with each other. It's simply all about utilizing different habitats. And the example that's given here are the two separate species of garter snakes that live in the Midwest. One is a dryland snake. The other one lives in more swampy regions and is perfectly happy swimming. These two snakes never meet each other because the habitats that they choose to live in are very, very different. So they live, they eat, they mate, they live out their lives in two separate habitats. They never encounter each other, they never mate. The next one is all about timing, the temporal isolation, the timing of mating. So here you can see two individuals that represent the two species of polecats, again, a Western species and an Eastern species. Polecats, you might think, could have the potential to mate. They certainly are fairly similar to each other. Their ranges overlap. Again, in the Mississippi Valley, everything is there with the opportunities for them to mate, with one exception. They mate at different times of the year. So one species mates at the beginning of winter, just as winter is setting in. The embryo goes into stasis, into suspended animation, and it remains in stasis within the body of the female until the beginning of spring, when the resources are available. Embryonic development kicks in again. The young are born early in spring to take advantage of the new food resources that are available, and normal development occurs from that point onwards. The other species does not mate until spring. And so what you find is that if an individual of one of these species who was ready to mate encountered an individual of the other species, that the individual from the other species simply would not be interested because it's the wrong time of year for mating. So timing is everything here. And if the timing is not right, if one of the two individuals is not ready to mate, then mating is not going to happen. The two species remain reproductively isolated because they're interested in mating at different times of the year. The next one is one that biologists, and particularly biologists that work to recover endangered species, have encountered, particularly with birds. And this is all about behavioral isolation. Birds are noted for the requirement of the mating pair to perform mating rituals in front of each other to acknowledge that they are members of the same species, that they are forming a mating bond or a pair bond for the purposes of mating. And each species has its own unique behaviors that establishes these pair bonds. Even in birds where the individuals mate for life, they still do these mating rituals to reestablish the mating bonds when they encounter each other again for the purpose of mating. So not all lifetime mating pairs actually stay together for their lifetimes. They may go their separate ways after they've finished raising the young. So this is the reason why they need to reestablish that pair bond. So where have biologists run foul of these? Well, one of the interesting things that happens with young birds is that when they emerge from the egg, as they look around at this world that they've emerged into, the first large moving object that they see in front of them, they fixate on. We say that they imprint on it, and they view that large moving object as their parent. Now, typically in nature, this is not a problem, because the parental birds, the birds that are responsible for those eggs, are the ones that the young see first and imprint on. And even in situations where you've got parasitic birds like the cuckoos, the young cuckoo imprints on the mating pair that are taking care of the nest and considers itself to be the young of that pair until it fledges, in which and then it leaves the nest and becomes a cuckoo and takes on cuckoo behavior. It's a slightly different behavior, but nonetheless. Anyway, getting back to the story. A number of endangered species of birds have attempted to be recovered to larger populations by human intervention. And the most famous story of all of this is with the whooping cranes that are very closely related to the sandhill cranes that we have here in Florida. There's a pair that use the college campus and the surrounding environs as their habitat. They're on campus right now with their chicks from this year. They've actually managed to raise 2 chicks to maturity. I see them out on campus when I'm walking around. This year, they seem to be a little bit shy of humans, but that may be because they've been used to having the campus all to themselves and not seeing a lot of humans. But under normal circumstances, with a lot of people on campus, they're relatively approachable. You can usually get within 6 feet of them before they'll turn around and walk away. Sometimes they'll even come over to take a look at you because they're a little bit curious. But what biologists realized is that if they take out an egg or two from a nest of a whooping crane, the pair will simply go ahead and produce more eggs. the female will lay a couple more eggs to replace the ones that are missing. If they then take those eggs, and this is where best intentions come into play, and place them in a whooping crane nest, the whooping cranes see those eggs, they consider them to be their own, and they deal with them as usual. When the chicks hatch out, the chicks see the whooping cranes, fixate on them, imprint on them, and consider the adult sandhill cranes to be their parents. And therein becomes the problem, because they now consider themselves to be sandhill cranes, even though they are whooping cranes. When those cranes reach maturity, they can no longer mate. And the reason is quite simple. The mating behaviors are instinctive. They're hardwired in the brain. They are not learnt by observation. And so these whooping crane chicks know how to perform as whooping cranes, but remember, they're imprinted on sandhill cranes. They think they're sandhill cranes, and they perform their mating dances with sandhill cranes. And the sandhill cranes look at them and go, huh, who are you? I don't recognize you. And so a whole generation of whooping crane chicks, which was not a lot because there's only about 500 whooping cranes in existence right now, unfortunately. A whole generation of chicks were raised that simply saw themselves as sandhill cranes and were no good for mating purposes because they would not mate with whooping cranes. That has been fixed now, and the population is increasing very slowly. They are even teaching the chicks to migrate, or the young to migrate along the historic migration pathways of whooping cranes, using ultralights and people dressed in white robes. That apparently seems to be all the young need to do to recognize that these are crane-like objects that they should follow. And so long as the humans have hoods over their heads so their faces can't be seen, the cranes will follow. They only need to follow the migratory pathway 1 year. The birds learn it, and they will follow that migratory pathway for the rest of their lives. So there is a migrating population that migrates down out of the Midwest to northern Florida, to some reserves up there that are reserved specifically for the whooping cranes. You can go up and visit, but you can't get near the birds when they're in residence. Everything is fenced off, so the birds remain undisturbed, but you can see them in the distance. And these are birds that are important to preserve because they are the tallest flying birds that we have here in North America by an inch or two. And there are a number of other stories that I could tell you about raising populations of birds that are on the brink of extinction, taking into account what we now know about imprinting and so forth, but that's a story for another day. So these are the three barriers that prevent a mating attempt. Now, of course, a mating attempt still does not mean fertilization is going to take place. Just because you can mate doesn't mean that there's compatibility between eggs and sperm. So onwards and upwards. So assuming that a mating attempt takes place, the next barrier is what is referred to as mechanical isolation. This simply means that the genitalia are incompatible. And the picture that's shown here, and the one that's used in the textbook, which we'll talk about a little bit more towards the end of the chapter, involves a pair of snails that are trying to mate with each other. The problem with these snails is that they belong to two different species, or at least they do now. But historically, they were members of the same species. And we know the origin of these two species because it's the product of a single mutation that caused the whorl of the snail shell. You can see the spiral pattern on the snail shells. It caused the whorl of these snail shells to oppose each other. And because of the way in which the snail body, the gastropod body, as you'll see when we do gastropods, developed, the genitalia are positioned as with respect to the shell itself and the opening of the shell. And the opening of the shell is actually offset either to the left or the right in these two species of snails. off to the left in one species, off to the right in the other species, which means the genitalia are in different locations, which means they cannot exchange sperm. They are hermaphroditic. They exchange sperm as part of their reproductive strategy. But because their genitalia, breed penis in this case, are incompatible, they can no longer mate with each other. And it was a single gene change that gave rise to this. Historically, we know the origin. So there could be a mechanical incompatibility. Well, what about a situation where you don't even have to carry out the mechanical practice of mating? What if you just release your gametes into the water, which a lot of marine organisms do? So echinoderms, and here you can see sea urchins, but you include sea stars and the like, corals. A lot of marine organisms do just release the gametes into the water. And you would think, well, under these circumstances, how do the right sperm and the right egg get together? How do you guarantee that only the correct sperm fertilizes the egg? And this is where sperm recognition proteins come in. We'll talk more about this when we do chapter 47, because we'll be talking about sperm compatibility and how the sperm makes it to the egg. But suffice to say that in these organisms, there are proteins on the surface of the egg that serve as receptors and recognize proteins on the surface of the sperm to identify the correct sperm from those that are incompatible. So these are sperm egg recognition proteins, and they must match in order for the sperm to be able to fertilize the egg. And so if the proteins are incompatible, then the gametes are going to be isolated from each other because the sperm cannot fuse with the egg. That's gametic isolation. So that's a barrier that would prevent mating if the mechanical isolation, the structure of the genitalia, did not prevent a potential mating event. So if all of these barriers fall down, and you know we've got 5 in sequence, if all of these barriers don't exist, then we have the possibility for fertilization, because there's nothing to prevent egg and sperm from uniting. That doesn't mean, however, that the individuals of the two populations represent the same species. Because remember, the definition is not just they reproduce or they produce offspring. These offspring have to be viable and they have to be fertile in their own right. So the first post-zygotic barrier, post-fertilization barrier, is the fact that the hybrids, the product of the mating, are themselves not viable. They don't survive to sexual maturity. They have physical deformities, they die as embryos, they die as young. They just fail to thrive and fail to reach sexual maturity in order to be able to reproduce. So if the hybrids fail, if they are not viable in their own right, if they cannot reach sexual maturity to mate with either other hybrids or with one of the two parents, then we have reduced hybrid viability and a barrier still exists between the two populations or the two species. If it is possible for them to produce offspring, but those offspring themselves are sterile and unable to reproduce, then we still have a barrier. This is reduced hybrid fertility. And this is what separates donkeys and horses. You should be aware, I hope, that donkeys and horses can breed and they produce mules. This is where mules come from. But it is not possible to produce a breeding population of mules by virtue of the fact that the male mules are sterile. They cannot produce viable sperm. And because they can't produce viable sperm, there's no way that a male mule can mate with a female mule to produce more mules. So you have reduced hybrid fertility, or you have zero hybrid fertility here because they're incapable of producing offspring. So horses and donkeys remain separate species, even though they can produce viable offspring. The offspring do reach adulthood. and what would under normal circumstances be considered to be sexual maturity. Then if all else fails, if the hybrids themselves can reproduce and produce offspring, if those offspring are themselves non-viable and non-fertile or reduced in viability and fertility, then you have what is referred to as hybrid breakdown. And that represents the final, granted, very small, but nonetheless very real barrier between the two species. If none of that affects the reproduction between the two populations, if they can produce viable fertile offspring that themselves are viable and fertile and leave behind viable and fertile offspring, then there is no reproductive barrier between the two populations. They are capable of interbreeding, and we consider them to be members of the same species. So you can see there are many, many opportunities for reproductive isolation to occur. And if even just one of these barriers exists between the two populations in question in nature, because remember that's where we've got to see this, then we have to treat the two populations as separate species. Could they be undergoing a speciation event, or could they be collapsing back into a single species? That would require further observation and further long-term observation to sort out that question. But if we observe at least one of these barriers in place between the two populations in nature, then we have to consider the two populations to be two separate species. So that's how we apply the biological species concept to populations and groups of organisms in nature. But remember, this is not a one-size-fits-all proposition. This definition cannot work in all situations, and here's just a few that may come into play. So we've already talked about asexual organisms. Implied in the species, the biological species concept is the notion of sexual reproduction. So immediately it cannot apply to asexually breeding organisms. You can't apply it to fossils, as I mentioned earlier, because we can't observe the mating patterns of dead organisms. And remember, we must observe their mating patterns in nature, which also means if we don't know very much about how they reproduce, and surprisingly, that is the case for a number of species, particularly species in the ocean where we may have only encountered them once, and we know very little about them other than they exist. And this is even true for a number of species that we know of in more isolated areas. So the western end of the island of New Guinea, what is known as West Irian, part of Indonesia, contains valleys that have not been penetrated by Western biologists at all. Just a few years ago, a couple of valleys were penetrated by Western biologists for the very first time in recorded history. They took with them individuals of the local tribes because those individuals did know the organisms that lived there. But Western science had not been in there, did not know anything that existed. The group that went in spent 2 weeks. God, you don't recognize what a fortnight is. That's a British term. It means 2 weeks. Spent 2 weeks in there and came out with about 30 new species of organisms, and they had not exhausted the number of new organisms they expected to find. There is plans to go back and take another look, to put together another expedition and go back in and take a look. But at this point, while we may be able to identify those organisms as potential new species, it's quite probable that they need some more work and, of course, a lot more observation in nature where it matters. So as a consequence of that, we do have other species definitions. We've got the morphological definition, which is the older definition. This would be the sort of definition that someone like Linnaeus or pre-20th century biologists would probably accept as a pretty good definition of a species. because it's based on observation of morphology and anatomy. When it comes to fossils, we can't even really focus on morphology because all we typically have is hard parts. We have skeletal parts. And we know that the external features are as much a part of mating selection as the internal skeletal features. And what about all of those organisms for which we have no hard parts? We may have evidence of their existence because we have imprints of soft tissue, but we don't have hard parts in order to be able to do more diagnostics. So there is another definition that is applied to those organisms that are extracted out of the fossil record. It has to do with location and the likelihood of different populations to be able to move around sufficiently that they would encounter each other and therefore mate. So if you have skeletons of two, let's say dinosaurs for the moment, two dinosaurs where the skeletons appear to be similar or near similar, but they're located on different continents, you could say, well, maybe they are two different species, but then we have to think, well, what about continental drift and plate tectonics? Were these continental land masses near each other during the time that these organisms lived? Was there possibility for migration between these two geographic locations in deep time? Lots of questions have to be asked, but nonetheless, people continue to do so. If, of course, two skeletons are isolated from the same location and they are morphologically similar, then they are considered to be the same species. No real possibility. Then we have an ecological species concept. This is applied particularly to bacteria, and it relies on their utilization of resources in the environment, hence the term ecological. Then you have the phylogenetic concept, which applies to organisms that share a common genetic history. So you look at their genes via gene sequencing. That may not necessarily be accurate, but at least we can use it for organisms about which we have very little information other than perhaps a genome. This is one of the reasons why we are really interested in the genome of Neanderthals. It's the reason why it was sequenced as one of the first human species to be sequenced other than Homo sapiens once the Human Genome Project was completed, to see if Neanderthals were very different from humans, a different species. Were they subspecies of humans? How were we related? Did we breed with them or not? And the answer is they were separate species. We are related to them. We did interbreed with them. We share a common ancestor. Back in the ancestral home of sub-Saharan Africa, there, the population that gave rise to them migrated out about 1 million years ago into Europe and Asia, probably gave rise to the Neanderthals and the Denisovans, and probably the Homo floresiensis, which occupied the island of Florence in the Indonesian archipelago. You may have heard of them as the hobbits. These are dwarf humans and real dwarf humans. But they may all have been derived from a population that was also ancestral to ours, but so they represent the first members of genus Homo to migrate out of Africa. Sometime before our own species emerged, and followed them about 800,000, 900,000 years later. And again, we mated with them because we know that their DNA is present in our genomes, and there's only one way that that could get there. So we do have these other species definitions, and as you can see, a number of these can be applied not just to sexually reproducing organisms, but also to asexually reproducing organisms. So this is why we can talk about species of bacteria, because we have some definitions that can be applied to asexually reproducing species and allow us to identify species that don't reproduce sexually. So that's some background to speciation. That gives you an idea of what we are looking for when we want to identify whether speciation is taking place or not. We look for the presence of these reproductive barriers, and we look to see whether those reproductive barriers are creating reproductive isolation. So that brings us to the end of this section. We're going to take a break, and as always, I have some concept check questions for you to think about. And I will close down this section and come back with a new section of recording and answer these questions. So let me read out the questions, then we'll take a break, and I'll see you on the other side of the break with the answers. So here's the questions. Which species concepts could you apply to both asexual and sexual species? Which would be the most useful for identifying species in the field? You find two species of bird that utilize different parts of the forest, treetops versus ground, for instance, for feeding and mating. In captivity, however, they produce viable fertile offspring. So they are capable of reproducing in the lab. What reproductive barrier keeps them separate as species in nature? And explain why it's that reproductive barrier. See you on the other side of the break for the answers. No, I'm not sure what's going on here. I'm trying to close down the recording. My mouse won't work. So. Oh, well, we'll just have to move on, I guess. I'm not sure why the mouse. I'm going to pull the slide across. I'm going to change the battery and see if that's the cause of this problem. Maybe the battery's run out. Right, a new battery in, and it's still not. Let me turn off. I don't know. All I can do is. Okay, I'm not sure how this is going to play out. I forcefully stopped the previous recording. If necessary, I can go in and edit it before I share it with you and cut out all of the junk. But if you see a little bit of junk at the end of the previous recording, I'm sorry about that. I'm not sure exactly what happened here, but let's move on. So here are the two concept check questions that I finished the previous section with. So let's take a look at the answers. Which species concepts could you apply to both asexual and sexual species? Which would be the most useful for identifying species in the field? Well, let's just go back. It's only a couple of slides back and take a look. So the phylogenetic concept we could use because it applies to both sexual and asexual species. The ecological concept we can use because it can apply to both sexual and asexual species. And we could use the morphological species concept. Which would be the most useful in the field? Well, not the morphological, because remember that's based on physical appearance, and appearance can be confusing, as we talked about at the beginning. So we probably wouldn't want to use this one because it could be kind of confusing. The phylogenetic species concept probably wouldn't work in the field because we don't have any evidence of shared evolutionary history by observation in the field. But we could use the ecological concept because we could clearly see whether they were using different habitats or not. And in fact, that leads us to the next question, because this second question gets to that point. You find two species of bird that utilize different parts of the forest, treetops versus ground, for feeding and mating. In captivity, they produce viable fertile offspring. So there is a suggestion, at least from the lab experiment, that they may be the same species. But they're not. Why not? What is the reproductive barrier that keeps them separate as species in nature? Remember, the definition, the biological species definition, says that the mating observations have to be in nature, not in the laboratory. So because they can produce viable fertile offspring in the laboratory doesn't mean they're the same species. But the hint is that they're utilizing different parts of the forest, treetops versus ground. They're utilizing different habitats. So if we wanted to pinpoint which of the reproductive barriers is in play here, it would have to be the habitat barrier. They're utilizing different habitats for feeding and mating. Therefore, they would rarely, if ever, encounter each other, in which case it is very unlikely that they would interbreed under those circumstances. And that's how we could use the ecological definition in the field to try and identify populations that really represent separate species by looking at the habitats that they're utilizing. So let's move on and continue looking at speciation by addressing the two basic modes of speciation. So the two modes of speciation are referred to as allopatric and sympatric speciation. Let me fire up the pen again. So allopatric speciation refers to different fatherlands, in other words, different geographical origins, And sympatric speciation translates as same fatherland, meaning speciation takes place in the same geographic location. So basically, we're talking about speciation as a consequence of being isolated geographically, or speciation taking place within the same geographic location. We'll take a look at allopatric speciation first, because that's the easiest to understand. Sympatric speciation is a little bit more complicated because we're looking at speciation within the same geographic location rather than separate geographic locations. So this diagram just simply illustrates what we're talking about here. Here you have a pond that, or a lake that is a single ecosystem for a while, and then the lake dries out a little bit, creating two separate habitats. The two populations of fish then become separate species over time as they remain isolated from each other. Or you could have changes in the lake or in the behavior of the fish. Something happens that causes the fish, to certain members of the population, to become reproductively isolated over time. We'll talk about some situations where this has happened, because this is actually played out in Africa. The Great Lakes of Africa have given rise to a significant number of species of fish that are known as cichlid fish, as a consequence of both of these two basic mechanisms being in play. At various times, the lakes have been united or been separated into separate lakes. When they've been united, different regions of the very large body of water have taken on different ecological characteristics, which has given rise to different species of fish. At other times, the lakes have reduced in volume and become separate bodies of water, which has created the opportunity for speciation through allopatric speciation. So we know that there are regions around the world where these two modes have been in operation at different times, depending on what has been happening ecologically and geologically. So let's take a look at allopatric first. So allopatric simply means loss of gene flow. Remember, that's what unites populations together, the flow of genes backwards and forwards between the two populations, and creates reproductive isolation. When you lose gene flow, you automatically have reproductive isolation. That is what is implied by reproductive isolation. So here we're talking about reproductive isolation as a consequence of some geographic event or geologic event that creates geographic isolation. So here's a classic example. There are two species of ground squirrels that live in the Colorado Plateau region, through which the Grand Canyon has been carved by the Colorado River. So you have a northern side and a western side. We talk about the south rim and the north rim of the canyon. But you've got a northern plateau and a southern plateau. And there are two species of ground squirrels that live in these two regions. And they are recognized as two separate species because the canyon represents a very real geographic isolation mechanism. It is over a mile deep. It is vertical in many places, and a little ground squirrel is simply not going to take the time to crawl down one side of the canyon, find a way across the river in some way, and then crawl up the other side of the canyon just to find a mate. It's much easier to mate within your own population on your side of the canyon. So for animals like the ground squirrels, this is a very real reproductive barrier. It is a very real geographic barrier. And so the rise of these two ground squirrels is a product of allopatric speciation. Should they ever encounter each other for some reason, they may still be able to reproduce, but it would be very unusual for them to encounter each other in the wild. Other species are not daunted by the canyon. So anything that can fly, birds, some insects, plants where the seeds are carried on the wind, or the pollen is carried on the wind, or the pollen is carried by pollinators like birds and insects and bats, may very well represent a single species on both sides of the canyon, because they are not reproductively isolated from each other by their own individual behavior or the behavior of those animals that they interact with for the purpose of reproduction. So what constitutes a geographic barrier for one species may not necessarily be a geographic barrier for other species. It depends. But nonetheless, what we are referring to here is reproductive isolation, loss of gene flow as a product of geographic isolation. Here's another example. So there are two pairs of sister shrimp here. These two, and fire up the pen again just to make sure that we get something. These two are a pair. What did I? Again. Oops. That's good. So these two represent a pair of sister shrimp, and these two pairs represent a pair of sister shrimp. And there are, in fact, 12 pairs of sister shrimp on either sides of the Isthmus of Panama. And you notice that the Isthmus of Panama actually runs east-west. And Panama Canal, which if you look very carefully, you can see the evidence of right there, actually runs north-south across the isthmus. Now, the reason why this represents speciation due to geographic isolation is because the Isthmus of Panama has not always been a connection between North and South America. For much of the existence of North and South America, they've actually been separate continents. Land bridges between them have come and gone, but the current land bridge, the Isthmus of Panama, emerged somewhere around 12 to 15 million years ago is what is generally considered to be the time frame. Some people talk about a little bit later in time, a little bit further back in time, but most people are of the opinion it's around 12 to 15 million years ago that the isthmus permanently connected North and South America as we understand it today. And so these two species of shrimp have emerged as a consequence of being isolated from each other by the appearance of the isthmus. And so this is a classic example of speciation via allopatric speciation, the geographic isolation of the two populations. Remember, there's another 10 sister pairs of shrimp species separated by the isthmus. So this is a very nice example of allopatric speciation. So the big deal about allopatric speciation is to demonstrate that reproductive isolation has occurred, that there is no evidence of gene flow or the possibility of gene flow. That's not always necessarily easy to demonstrate, but here's a series of experiments that were done to demonstrate how quickly isolation or a reproductive barrier can emerge as a consequence of geographic isolation. So a population of Drosophila, Pseudo obscura is a relative of Melanogaster. That's the little fruit fly that we talk about in genetics. A single population of this Drosophila that had been raised on a sugar source was then divided into two. One of the two populations was then raised on starch. The other was raised on maltose as the sugar source, and were kept separated from each other for about a year, which is approximately time enough for 40 generations. Generation time for fruit flies being approximately 2 weeks. After 40 generations, the flies were then reintroduced to each other. But prior to being introduced to each other, they were placed on a cornmeal food source to clear out any evidence of the sugars. So there was no evidence of the sugars in the digestive tracts of the flies so that they could sense the presence of a foreign sugar in their mating partners. Then the flies were matched up on the basis of same sugar source versus different sugar source and given opportunities to match. And you can see the results of the experiments here. So where they were matched up with individuals of different populations grown on the same sugar source, here and here, there was no preference shown for mating partners. They remained mated very strong. They mated very strongly. Well, there was preference shown, sorry. They mated very strongly with members of their own that had grown up on their own sugar source. Occasionally, very small number, mated with individuals that were grown on the other sugar source. But the vast majority of flies chose preferentially to mate only with members grown on the same sugar source that they were grown on. A separate population was maintained on starch. Multiple populations actually were maintained on starch over the same period of time and were again allowed to mate with either members of the same starch-grown population or members of different starch-grown populations. And you can see here, there's no real mating preference. They were perfectly happy to mate with members of their own population versus members of different starch-grown population. Remember, this is in a space of 40 generations. There's already a reproductive barrier emerging on the basis of selectivity, on the basis of what sugar source the adults were grown on, or the flies were grown on. They could already detect some difference. Now, we don't know whether this is the ability to detect the actual sugar source that they were grown on, or whether this is simply a product of other factors that were arising that created a reproductive barrier as a consequence of being isolated and grown on different sugar sources. It's not clear from the studies whether this is a direct reaction to the sugar source that they were grown on, or whether this is the product of other changes that led to mating preference as a consequence of being grown on different sugar sources. But the data from the starch, the populations that were grown on the same sugar source over the same length of time, but when compared with those that were grown on different sugar sources, suggests that it is a reaction to the sugar source, that somehow they are able to detect individuals grown on the same sugar source versus individuals grown on different sugar sources. That it's not a consequence simply of the time that they've been separated. It is a product of the environment that they were living in and the sugar source that they were utilizing. That may have caused some change, but it's very difficult to sort through that one. There are a whole lot of variables that could be in play there, and the authors of the paper did not speculate on that. Sympatric speciation is something that is a little bit more difficult to understand because it relies on loss of gene flow, even though the two populations occupy the same geographic location. So as you can imagine, the reproductive barriers here are going to be more definitive. It can't be just the product of physical separation anymore. There has to be some very clear-cut reproductive barriers between the two populations that lead to speciation. So while in animals, it's a little bit hard or a little bit difficult to identify very clear-cut natural situations of sympatric speciation, where we can genetically identify the situation. In plants, it's a little bit more straightforward. Plants tend to be a little bit more genetically forgiving when changes in chromosome numbers occur. So the situation is known as polyploidy, and polyploidy refers to the fact that there is a non-disjunction event during meiosis that leads to the failure to separate amongst all of the homologous pairs of chromosomes during anaphase 1 of meiosis 1. That means that the resulting gametes wind up with all of the copies of chromosomes. And so the gametes themselves are diploid. And you might say, well, how can that happen? Well, we know that non-disjunction happens. How can a species reproduce if its gametes are no longer chromosomally compatible with its parents? Well, there are two basic mechanisms where this can under which this can happen, certainly in plants. So the first situation is referred to as autopolyploidy. This occurs within a single species or individuals within a single species, and it is very common in plants. We know that this happens. We've seen it happen in the laboratory, and we can identify it in nature. So you have the non-disjunction event, which gives rise to tetraploid cells. In this case, the error is occurring during normal cell division, which leads to a tetraploid cell that then undergoes meiosis. It could go either way, but the result is that you have gametes that wind up being being diploid. Those are incompatible with the haploid gametes of any of the surrounding plants. But if this plant is capable of self-fertilization, as up to 50% of all plants are capable of, then this plant could reproduce with itself. Its own eggs and sperm could fertilize each other and give rise to fertile offspring, simply because while the gametes may be incompatible with the surrounding plants, they are compatible with each other. And you immediately see the emergence of a new species in one generation. And we know that this has happened multiple times within the plant kingdom. We know that something like a similar sort of process can happen with our agricultural plants. More on that in a moment. But let me tell you a little bit about the other form of polyploidy. So this is autopolyploidy that occurs within a single individual plant. Allopolyploidy requires multiple species typically to create a hybrid that is a chromosomal fusion, a hybrid of the two parentals that then becomes fertile itself, but is no longer capable of reproducing with either of the two parental species. And again, you may ask, how can that happen? Well, here's this circumstance under which it might happen. The reason why we can say that this is a way for for a polyploid plant to occur is because this is the way in which, for instance, our domestic wheat emerged. Again, more on that in a moment. So it requires two closely related species that produce normal gametes that are haploid, that are compatible with each other. There is no block to the gametes fertilizing each other. Now, as you'll see when we talk about the plant kingdom, there is pollen incompatibility protection in some cases to prevent self-pollination, specifically to prevent self-pollination, but also in some cases to prevent accidental pollination by pollen from other species, pollen representing the sperm in plants. But if there is no block to the pollen fertilizing the egg, and because the gametes are haploid, you now create a hybrid that is itself infertile because it has no matching homologs to create homologous pairs of chromosomes. So you might think, well, that's the end of this. But if it is able to vegetatively reproduce from cuttings, produce new individuals by asexual reproductive mechanisms, which a lot of plants are capable of doing, either through cuttings, by budding. There are a number of different mechanisms that plants can asexually reproduce. This otherwise sterile hybrid, sterile in terms of sexual reproduction, can now create extra individuals. And so it can create a new population of individuals that are reproductively isolated from both of these parents, but it's still not capable of sexual reproduction. But somewhere down the line, it appears at some point, there are errors in cell reproduction, either at the mitotic or meiotic level, that give rise to diploid pairs of chromosomes that creates now the possibility for meiosis, and they can start reproducing sexually and create a new species of sexually reproducing plants rather than just something that's reproducing asexually. And so it introduces sexual reproduction into the life cycle of this new species. And this, we know, is the likely mechanism that gave rise to domestic wheat, because domestic wheat happens to be an allopolyploid. It happens to be hexaploid with 6 complete sets of chromosomes. And I'll show you a diagram in a moment that illustrates how we think domestic wheat came about. But here's an example of 2 new species arising as a consequence of hybridization between 2 plants. In this case, the parental plants, and there are 3 of them, all have 12 chromosomes, but their hybrid offspring all have 24 chromosomes, and they are the fusion product of the top species hybridizing with one of the 2 species at the bottom. These are plants that grow in the Pacific Northwest. They are introduced species that have in turn become adapted to the Pacific Northwest, but in the process of adapting have in turn given rise to new species in regions where their ranges overlap. And so there are now endemic new species that are the hybridization product of these 2 parental species. Something's going on with my computer. The top screen just changed, and yeah. Well, I'm just going to have to stop it the way I stopped it last time when we get to the next break, which is coming up. So here are some agricultural plants that we know are polyploid, and you can see here that these are all domesticated plants, and either directly domesticated from wild relatives or are the product of some breeding. Let me show you wheat, because we know the origin of wheat is a hexaploid. So here's the diagram of the 3 wild relatives of wheat, and you can see that each of them has 14 chromosomes. And then if you multiply 14 by 3, you get 42, and that gives you the hexaploid number for domestic wheat. And we know that these are the chromosomes that have come from the wild parents. These wild parents still grow wild in the Middle East where wheat emerged, probably in part as a consequence of hybridization in nature, but also in part as a product of probably some human intervention, whether it occurred to give rise to some of the hybrids, or whether human intervention only took place after the hexaploid ancestor of modern wheat emerged is not clear. Wheat as a domestic crop has been grown for around 12 to 13,000 years. So the records from that time period are a little bit sketchy, so we don't know exactly how wheat as a hexaploid directly emerged, but we know that it was a product of hybridization between these 3 wild grasses. So one of the other things that can give rise to sympatric speciation is mating preference, sexual selection, particularly on the basis of females, choosing males on the basis of particularly of physical appearance. And as you might guess, this is a type of non-random mating, as the note suggests. This has been demonstrated in the laboratory that this is a very real effect. And as you'll see later in the presentation, we're actually starting to observe the very non-random mating occurring in nature that has been identified in the laboratory. In fact, it was the possibility of non-random mating in nature as a product of changes in the environment that prompted these experiments to be done. So let's take a look at the experiments. So what you have are two species of cichlid fish that live in one of the large lakes in Central Africa. So you've got Pundamelia and Nyeriae, and in normal light, they are easily distinguishable between each other. At least the males are easily distinguishable between each other. So if the water is clear, there's no turbidity, there's no debris and other materials in the water, the females can clearly distinguish between the males of their own species and males of the other species. But introduce a physical change that makes the two males indistinguishable from each other. In this case, instead of using white light, using orange light only, the females can't distinguish the two males from each other, and they randomly mate with either of the two males. And that gives rise to a hybrid that is clearly a combination of the two morphological characteristics of the two species. There is actually a male hybrid that is a blend of these two in terms of physical appearance. And that we have seen being produced in the laboratory. What is interesting is, as I said, and we'll talk about this a little bit later in the presentation again, we're seeing this starting to emerge in one of the lakes in Central Africa, where these two species may be starting to merge into each other because of the loss of the loss of ability to differentiate between the two males. And there are some reasons to explain why, which I'll get into when we come back. So by way of summary, allopatric speciation and sympatric speciation can be differentiated from each other in the following ways. Allopatric speciation requires geographic isolation, where the two populations are geographically isolated from each other. The two populations then evolve in different directions as a consequence of accumulating adaptations to their environments. When they meet again, if they should ever meet again, they are reproductively isolated from each other in some fashion other than geography. In sympatric speciation, it takes place within the same geographic location. It requires reproductive barriers of the forms that we saw at the beginning of the presentation. So those reproductive barriers that are either anything other than habitat isolation, basically. Any of those subsequent barriers that prevent either mating or fertilization or lead to hybrid breakdown, any of those would represent reproductive barriers that would come into play in sympatric speciation. But some of the earlier reproductive barriers would be stronger barriers in a sympatric speciation than perhaps some of those later barriers, more fuzzy barriers post-fertilization. So I want to take another break here. I guess I'm just going to have to shut this down manually somehow. We'll see how we get on. So here is my concept check for this break. Summarize the key differences between allopatric and sympatric speciation. Which type of speciation is more common and why? Is allopatric speciation more likely to occur on an island close to the mainland or on a more isolated island of a similar size? So I'll see you on the other side of the break with the answers. So I can't see the window right now, but I'm going to assume that the window is, let me just try something. Okay, so there's the window again. Yeah, there is something not right here somewhere along the way. So we're going to start up. This is where we finished off. Yeah, I'll do it this way. I'm going to have to take another look at my computer after we're done and see if I can find out what on earth is going on here. Anyway, we are back recording again, so let's keep going, see what we can do here. So here are the concept questions that we finished up with at the break. So let's get the answers here and then we can continue on. So summarize the key differences between allopatric speciation and sympatric speciation. Which type of speciation is more common and why? Well, remember, allopatric speciation occurs due to geographic isolation. The two populations become geographically isolated from each other, interrupting gene flow and leading to speciation as a consequence of accumulation of unique adaptations, unique mutations in each of the two populations. Sympatric speciation is a loss of gene flow due to reproductive barriers other than habitat isolation or geographic isolation. So which of the two speciation events is more likely? Allopatric speciation, because geographic isolation is much easier and more frequent than the emergence of reproductive barriers of the type that we talked about in the first section of the presentation. So that's your answer to this question. Is allopatric speciation more likely to occur on an island close to the mainland or on a more isolated one of a small size? Always on the more isolated one. And the reason is fairly simple. If the island is close to the mainland, there's always the possibility of gene flow from the mainland out to the island. The closer the island is to the mainland, the more likely individuals are to be able to move from the mainland out to the island, either forcibly or voluntarily. And that gene flow as a consequence of that movement tends to dilute out any of the adaptations to the environment of the island. The further the island is away from the mainland, the less likely there is to be gene flow until such point you get far enough away that gene flow stops. And now you really do have geographic isolation leading to reproductive isolation. So allopatric speciation is always going to be more likely on an isolated island rather than on an island close to the mainland. So the next thing to talk about is what happens when two species share a common range, what is referred to as a hybrid zone. Hybrid zone is where not only do the ranges of the two species overlap, but where members of the two species encounter each other and mate to produce hybrids. And it's what happens in the hybrid zone that determines whether the species remain separate from each other or merge with each other again. It's the nature of the hybrids and what happens to those hybrids that determines whether the reproductive barriers between the two species are going to remain intact or are going to collapse. So let's take a look at a hybrid zone. This is one that's mentioned in the textbook. This hybrid zone is between the yellow-bellied toad and the fire-bellied toad. Now, the hybrid zone itself is about 4,000 kilometers in length. You can see that it winds its way through Central Europe. But the hybrid zone itself is only about 10 kilometers wide, which for a hybrid zone is relatively narrow. It's actually very narrow. So this is the range over which these two species of toads are likely to interact with each other. And the reason why this range is so narrow is because of the habitats that the two toads occupy. So the fire-bellied toad tends to live on the lowland, flat areas, whereas the yellow-bellied toad prefers more elevated locations. And so there is a narrow region where the yellow-bellied toad and the fire-bellied toad are comfortable living. And so this is where you have the potential for the hybrid zone. So the question then becomes, what is going on in the hybrid zone? And the way you can interpret this data is to look at the frequency of hybrids, because this is what the the frequency of yellow-bellied toads is in the population. So not surprisingly, on the left-hand side, at some distance from the hybrid zone, it's all yellow-bellied toads. No evidence of hybrids at all. As you get closer to the hybrid zone, you start to see the presence of some hybrids, but not a lot. The incidence of hybrids is relatively low. The incidence of yellow-bellied toads is still over 90%, right up to the hybrid zone. Within the hybrid zone, then you see a rapid decrease in the incidence of yellow-bellied toads. Now, all the individuals in the red hybrid zone represent the hybrids, evidence of yellow-bellied plus red-bellied genetics. So these are the hybrids, but you see that as you progress across this very narrow hybrid zone, the incidence of yellow-bellied toad contribution drops very, very dramatically. So it is less than 10%, it's down around 5% by the time you get to the other side of the hybrid zone. And then as you move rapidly, as you move away from the high edge of the hybrid zone into the red-bellied habitat, you see, again, a rapid drop in the incidence of yellow-bellied contribution. So why is there very little evidence of gene flow across the hybrid zone if hybrids exist? It has to do with the nature of the hybrids. The hybrids have very poor hybrid survivability. They have very high incidence of embryonic morbidity. They die as embryos. If they survive to emerge as toads, they have a lot of physical defects that limit their ability to reach sexual maturity. And if they do reach sexual maturity, they have very little opportunity to mate with individuals on either side of the hybrid zone. So there's very, very, very little opportunity for gene flow across the hybrid zone because of the breakdown of hybrid viability within the hybrid zone itself. So this, the data on this graph illustrates the breakdown of the hybrid. So just to highlight again what I was referring to, you can see over here that you have a very high incidence of the yellow-bellied toad contribution to the individuals, very little red-bellied toad contribution until you get right up against the edge of the hybrid zone. Over here, you have very little contribution from the yellow-bellied toads. It's largely fire-bellied toad, less than 5% contribution, even right on the edge of the hybrid zone. And then within the hybrid zone, you see a rapid loss of yellow-bellied toad contribution because of the poor survivability of the hybrids. They're just not viable. They have very low viability, meaning they have very, very low fertility. So there's not a lot of opportunity for gene flow from one side of the hybrid zone to the other side of the hybrid zone. So that's one example of what can go on in a hybrid zone. There are basically three things that can happen in the hybrid zone. You can see a strengthening of the reproductive barriers, a loss of gene flow or a reinforcement of the lack of gene flow across the hybrid barrier. you can see a breakdown of the reproductive barriers. In other words, you see an increase in gene flow through the hybrid zone, or you see a stabilization of the hybrid zone as a consequence of ongoing encounters between the two species that doesn't lead to a significant amount of gene flow, just a production of hybrids. And in fact, that's what's going on in the toad example. It's actually an example of a stabilization of the hybrid zone. It continues to exist because the members of the two species encounter each other and continue to produce hybrids. But because of the lack of viability and fertility in the hybrids, you just get an ongoing production of hybrids, but you see no evidence of gene flow across the hybrid zone. So the toad example is actually a really good example of stabilization. So I'm going to change this and say, and put a note in here, hybrid zone, stable basis. So that's what's happening when you get a continued formation of hybrids. So this diagram simply illustrates the three ideas. So you start out with a single species made up of multiple populations. One of the populations gets isolated because of a barrier to gene flow. It could be geographic, it could be one of the other barriers, so it could be allopatric or sympatric speciation, it doesn't matter. What you've got is a barrier to gene flow, so the population becomes isolated. The populations that are in contact with each other continue to have gene flow between them, keeping the allele frequencies more or less equal, so there's no differentiation between the populations, they remain part of the same species. Meanwhile, the isolated species starts to diverge as it accumulates its own unique set of mutations and adaptations. When you then get a coming together of the two species within the hybrid zone, then you have the three possibilities. You have reinforcement, which strengthens the barriers. You have fusion, where the barriers collapse and the two species merge back into each other again. Or you have the stability situation where you have an ongoing production of hybrids, but because of the hybrids do not promote gene flow, all you get is a constant production of the hybrids. The two species remain separated from each other. That's the stabilizing situation. So let's look at some examples of each of these. So if you want to look for strengthening of reproductive barriers, the place to look is for situations where sympatric speciation has been the mechanism of speciation rather than allopatric speciation. And there's a good example of a comparison that we can do between the strength of the reproductive barriers where speciation has occurred within the same group of species, and that's in Europe amongst the flycatchers. And this is documented in the textbook, so you can take a look at this in more detail if you wish in the textbook. But there are two basic groups of flycatchers in Europe. There's a group of species that has emerged as a product of sympatric speciation, and the males are clearly delineated because of their color patterns. So the females have no problem delineating or differentiating between the males. So when you look at the mating choices of the females, very clearly the females show an absolute preference for males from their own species and no preference for males from the alternative species. In other words, the reproductive barrier here is incredibly strong, but that we would expect because they are the product of sympatric speciation. The barriers have to be strong because these are birds that are living in the same habitat and are constantly encountering each other. So the only way to keep them separate as species is to have an exceptionally strong reproductive barrier. In the case of the flycatchers that emerged because of geographic isolation, allopatric speciation. When the females encounter males of the same species versus different species, when they have an opportunity to be selective, they show somewhat elevated preference for males of their own species, but they show a fairly significant tendency, I guess, to choose mates of either species. In other words, they'll mate with males of the alternative species simply because the difference between the two males in terms of color is very, very subtle. It's an elongation of the white patch here under the throat that differentiates the two males of the two species. And so the females, as a consequence, show some preference for the males of their own species because they can identify males of their own species. But encountering a male of the different species, they're not particularly picky because it's a little bit harder to differentiate between the two males, and the reproductive barriers between them are not particularly strong. They became separate species because of geography, not because of a strong reproductive barrier between them. They were just separated because of geography. So this is a circumstance where you see a reinforcement of the reproductive barriers where the species were created through sympatric speciation, because reproductive barriers here are incredibly strong. They have to be. In the case of weakening reproductive barriers, well, this is the chickweed fish of Africa, where we're starting to see gene flow between the two species as a consequence of the female's inability to be able to differentiate between the two males of the species, or the males of the two species, because the water in the lakes are becoming more turbid. Now, some of this is because of pollution. Some of it is because of agricultural runoff. Some of it is simply because of runoff of the soil into the lakes. It turns out that the soils are very volcanic. The Rift Valley itself in Central Africa, where these lakes exist, is very volcanic. And when that rock breaks down to soil, it gives a reddish orange soil, very characteristic reddish orange soil. When that gets washed into the lakes because of natural runoff or because of poor agricultural practices, the water turns very turbid. It turns a very orange red in color, much like the monochromatic orange environment in the laboratory where we saw the breakdown of sexual selection. This is actually starting to occur in nature, and we are starting to see the appearance of these hybrid fish that are clearly a hybrid between the two males of the two species, the males of the two species. And this is a clear indication that the reproductive barriers are breaking down, that these species may be in the process of merging back together again into a single species with the concomitant loss of the two parental species. A new species will emerge. That is the hybrid product of the two parentals, and the hybrid will become a new species that will ultimately become the dominant new species if the breakdown is complete. You may wind up with the complete loss of both species, or with the loss of one species and replacement by the hybrid. Which way that goes remains to be seen, but nonetheless, we are starting to see this breakdown between the two species and the appearance of these hybrids. In terms of stability, well, we've already talked about an example of the stability. That would be the hybrid zone between the two toads that we were talking about earlier, the yellow-bellied toad and the fire-bellied toad, where the hybrid zone is a stable zone because of ongoing reproduction between the two species, but because the hybrids lack viability and fertility, there's no gene flow to break down the barrier and there is still the ongoing production of hybrids, so we're not seeing a reinforcement of the separation. So we've got a stable hybrid zone going on. If something should change so that the hybrids increase in viability and fertility and actually have the ability to mate with one of the two parental species, then that could lead to a loss of the hybrid zone or a reinforcement of the hybrid zone. It depends on which way it goes. But for now, it's a stable situation and probably an ongoing stable situation for the time being, at least. So another break. And I think this time I'm just going to keep going because we haven't been going for very long since the last break. So we can just stop for a moment to take a look at the concept check questions and then continue on, because we've only got a few more slides to go to finish this presentation. So let's just keep going. So the concept check question this time is, what are hybrid zones and why are they viewed as natural laboratories in which to study evolution? Well, of course, hybrid zones are geographical regions where the ranges of two species overlap, and individuals from the two species can mate and produce hybrid offspring. So that's a hybrid zone. Why are they natural laboratories of evolution? Because here we can actually study evolution in process, because we can study the flow of genes between the two populations within the hybrid zone. We can see the nature of the reproductive barriers and the effect of those reproductive barriers on the ability for gene flow to occur. There are a lot of observations that we can make in a hybrid zone that we can't do in the laboratory, because remember, our species definition, our biological species definition, says that all of our observations have to be done in nature. So the hybrid zone is a perfect place in which to do all the observations that we need to do and let nature do the experimentation and just simply observe the results of nature's experimentation within the hybrid zone. So it's a perfect situation for evolutionary biologists that are interested in studying the process of speciation. So one of the issues that comes out of understanding that speciation can be documented over time, and that speciation is typically going to take place over longer periods of time, although our opinions are changing, obviously, then humans have the capacity to observe. The only way that we can look for a measure of how quickly speciation takes place is to look in the fossil record. Now, we can get some window on the rate of speciation by looking at gene sequences, but back in the 1970s and 1980s, prior to the development of automated sequencing, that sort of data didn't exist. But a glance at the fossil record and a understanding of what the fossil record is actually saying about the appearance of new species and their time in the fossil record before they become extinct tells us that the Wallace-Darwin view of gradual speciation is not well supported in the fossil record. If you assume that the average time, and this is a fairly accurate time period, the average time of a species in the fossil record is about 3.5 million years. The time span over which the appearance of the fossil or of the species in the fossil record takes place can be measured in tens of thousands, maybe 100,000 to 150, maybe 200,000 years, which, relatively speaking, is a very, very short period of time compared to the average time a species exists in the fossil record, or in terms of geologic time in general. And then it appears, at least from the fossil record, that the species remains relatively stable over the rest of its time in the fossil record until it becomes extinct. In other words, Speciation does not take place over long periods of time. It occurs very rapidly, and then the species remains relatively stable, or at least in terms of what the fossil record provides us with. Now, remember, the fossil record only provides us with hard parts like skeletons, teeth, things like that. It doesn't tell us whether there are changes in behavior. It doesn't tell us if there are changes in reproductive behavior. That doesn't tell us if there are changes in morphs, physical appearance. It just tells us about changes in the hard parts. And the fossil record suggests that changes in hard parts are very rare. Once the species is established in the fossil record, it becomes relatively stable, or at least that's the nature of the fossil record. So when Niles Eldredge and Stephen Jay Gould, who were both located at Harvard, at least Gould was at Harvard, I think Eldredge was also located at Harvard, took a look at this, they said the gradualist approach simply cannot be the way in which speciation occurs. A better way of looking at it is to talk in terms of punctuated equilibrium. The punctuated refers to that rapid period of time during which the species emerges in its mature form. And then the equilibrium refers to that long period of time where it remains relatively stable in the fossil record with very few changes. Just as an aside, we were very lucky. Shortly after I joined Valencia, to actually have Stephen Jay Gould come down and spend a day with us on campus and give a seminar on evolution. He was one of the most famous evolutionary biologists of his day. He unfortunately passed away about 10 or 15 years ago, but he left behind a huge volume of work in the form of popular science books talking about the nature of evolution and giving many, many, many examples of situations where evolution is in play, but where people are misunderstanding the nature of evolution and therefore misunderstanding what's actually happening. So if you want to read some interesting books, they're well written and easy to read. I would recommend getting one or two of Gould's books. One that I remember is The Panda's Thumb, talking about the notion of the panda having a thumb. It doesn't actually, it has an extension of its wrist bone that functions much like our thumb for feeding purposes. And I believe he was the author of a book called The Mismeasure of Man. It's another famous book on evolution and misunderstandings of evolution. I think Gould was the author of that book as well. If you're interested in getting a hold of some of these books, send me an email and I'll give you a list of some books that you can look up. I've got a number of these books and I can easily generate a book list for you, or you can generate a book list of your own. You don't need to go far, you can Google it. So this diagram illustrates the differences between the gradualist model and the punctuated model in terms of the appearance of new species. So in the gradualist model, you see this accumulated change over time with many intermediates, and each of these lineages of intermediates leads to the final species that appears in the fossil record. And you may, if you're lucky, be able to find evidence of those intermediates as part of the fossil record. In Eldredge and Gould's model, the punctuated model, the appearance of the new species occurs very, very quickly, relatively speaking. And then the two species remain pretty much stable at equilibrium in the fossil record throughout the rest of their time in the fossil record. Can we make the case for both? Sure. And the gradualist model is not necessarily wrong. It's just that based on what we see in the fossil record, the punctuated equilibrium model is a better explanation for the origin of species in most circumstances, not all circumstances, but in the majority of circumstances. It doesn't mean that Darwin and Wallace were wrong in proposing gradualism. They were simply proposing a model on the basis of the data that they had available to them at the time. They did not have the wealth of fossil material that we now have. They did not have the lineages of fossils that show us the progressive evolution of groups of animals and other organisms. I'm sure if they had the evidence that we have today, they would have talked in terms of something more along the line of the punctuated equilibrium model rather than the gradualist model. We can't fault Darwin and Wallace for proposing the gradual model. It made perfect sense in light of what evidence they had available. We just have better evidence. And so we can talk in terms of the punctuated model, but we can also talk in terms of the gradual model as well. Don't think that it is not a model for some types of speciation. It most certainly is. But it is not necessarily the only model anymore. So once we recognize that notion that speciation can take place extremely quickly once divergence starts to take place, we then have to think in terms of, well, how long between separate speciation events is there? Because one of the things that we see is that that interval can be incredibly short in the case of the cichlid fish, where we have constant changes in those large lakes in Central Africa. The speciation events may be occurring as quickly as 1 every 4,000 years, the emergence of a new species. Whereas in beetles, the speciation time has been quite long. Once the beetle, a new species of beetle emerges, it may be many millions of years before another species of beetle will emerge, with an average time of around 6.5 million years. How can we rationalize or correlate the fact that speciation appears to occur in very, very short periods of time, but there are long periods of time between separate speciation events? And the answer is, you have to remember what the time period between speciation events actually represents. It represents the combination of the time period during which the species are diverging from each other, which can be a relatively short period of time, plus the time it takes for another divergence to actually begin to lead to a new species. It's the combination of those two time periods that represents the interval between speciation events. So while speciation itself, the divergence of two populations to give rise to two new species, may be very, very rapid, very short periods of time, relatively speaking, the time it takes for a new divergence event to begin can be quite long, leading to long time periods between individual speciation events, but very short time periods over which the actual speciation process itself occurs because of the rapid rate at which divergence occurs. There's no contradiction here. They're actually mutually supportive. Once you understand that the time period between speciation events is actually the sum of the time that it takes for speciation to occur, the divergence event, plus the time it takes for a new divergence to begin. It's no more complicated than that understanding. So it turns out that speciation in nature can actually occur quite rapidly. So we know that the emergence of polyploid plants through autopolyploidy or allopolyploidy can occur relatively quickly. In the case of allopolyploidy, overnight almost, in the space of a single generation. But even when hybridization occurs, a new species can emerge relatively quickly. So what you're looking at here is a new species of sunflower that has emerged on dry sand dunes. And it is in fact a hybrid and it uniquely is able to occupy these dry sand dunes that the two other native species of sunflowers simply cannot occupy. It's too dry and sandy for them. They like a moister, more soil-based habitat. But this new species that is a hybrid of two existing species is able to occupy a new habitat. And it is expanding its range as it does so over these dry sand dunes. And some researchers at the University of Michigan decided to take a look at this because the ancestral species that gave rise to this new species were suspected. And so they could bring those two species into the laboratory and create artificial hybrids. And then could take a look at what happened with those artificial hybrids to see if they could monitor any changes that were occurring in those hybrids as they started to reproduce, because it turns out that the hybrids can reproduce and produce viable fertile offspring, and see if they could track any chromosomal changes that were occurring in the artificial hybrids and compare those with the known chromosomes from the naturally occurring hybrids. So the next slide, this slide shows you the comparisons between the native chromosomes, and we're only looking at 2 chromosomes here, so let me fire up the pen again. So H. anomalus is the experimental hybrid. Annuus is one of the 2 parentals, the native parentals, and Petiolaris is the actual name, is the other native parental. So in the laboratory, they created these hybrids, and initially the hybrids contained chromosomes that uniquely contained DNA derived either from Annuus or from Petiolaris. Now, it was known that in Anomalous, the chromosomes contained DNA in different regions that were either originated from one parent or the other. So the chromosomes actually contained a mixture of DNA from the 2 parents, but in different locations. Only DNA from one of the 2 parents existed. Incompatible DNA from the other parent apparently was no longer present, if it had been present at all. Well, in the case of the lab hybrids, it appears that the parental DNA probably was present at some point. But as the researchers tracked these F1 hybrids through 4 generations, so these changes occurred in as little as 4 generations. What they noticed was that regions of incompatible DNA were eliminated in each generation, so that the chromosomes that they were following, these 2 chromosomes that you can see pictured here, started to take on the characteristics of the chromosomes from the natural hybrids. And you can see by these comparisons, the colored regions between the 2 chromosomes, you can see that in these comparative regions, the chromosomes are almost identical to the chromosomes of the naturally occurring hybrids, this new hybrid species, Anomalous. There are still some subtle differences in chromosome 2, but it is definitely moving in the direction of the chromosome from Anomalous. And so it would appear that this new species of sunflower, this hybrid species of sunflower, may have emerged very, very rapidly in as little as 4 or 5 generations, apparently. Probably a little bit longer than that. But the one piece of information that I haven't given you about Anomalous that I need to give you now is that Anomalous is itself reproductively isolated from both Annuus and Petiolaris. It is most definitely a new species as a product of a hybridization event. And given that, at least at the laboratory level, where no intervention was used after the creation of the F1 hybrids, the emergence of the new species in nature may have occurred very, very rapidly. 4 to 5, certainly probably less than 10 generations. So a very rapid emergence of new species. But we've already noted that the emergence of new species in plants is not unusual. But it's interesting that in a hybridization event like this, you can get the emergence of new species. It takes a little bit longer, but nonetheless, you get the emergence of new species. So one of the other things that has been of interest, again, because of access to DNA technology, is what is the underlying genetics of speciation? How many genetic changes need to occur to start species or populations diverging from each other in the direction of new species? So the research that's discussed in the book is about some flowers, but again, I want to remind you that those snails that we were looking at back when we were looking at the reproductive barriers exist because of a single mutation in a single gene. Now, it just so happens that the gene involved is a master control gene, a Hox gene, that I've already introduced. No, I haven't introduced you to those yet, but I will introduce you to those in much more detail as we move through the semester. At least I don't think I've introduced you to them yet. I may have. It's a little fuzzy right now. But you can see here quite clearly what I was referring to when I said that the whirl, or the rotation of the spiral of the snail shell running in opposite directions. So just to reinforce that, I think I've got the pen up. Let's try it. Yeah. So this one is rotating in this direction. And the opening of the shell is pointing back this way. This one rotates in this direction, so it's obviously going in the other direction. But you also have to note that the opening of this shell is also facing back behind the shell. So they really are different. They are structurally different, which means that they are functionally different, which means they are mechanically, reproductively isolated from each other. And again, this is the product of a single mutation in a single gene. So even in animals, we have evidence for speciation occurring relatively quickly, very quickly. These are Japanese snails, by the way. So here's the experimental work that was done to try and get a handle on just what level of change needs to take place in order for a speciation divergence event to begin. So we have here two species of Mimulus. So this is species number 1, this is species number 2. Why we're focusing on these two is because they attract different pollinators. So species 1 is insect pollinated, specifically bumblebees. Species 2 is bird pollinated, specifically hummingbirds. Hummingbirds show a great preference to flowers with a red color. So there are a number of genes that are responsible for determining the coloration of these flowers. But the researchers focused in on a single gene and took these two species of Mimulus into the laboratory and created hybrids between them. It wasn't easy, but they were able to create hybrids between them. Once they had an F1 hybrid, then they started to back mutate the hybrids with the two parentals, and eventually were able to transfer the gene of interest from species 1 into species 2, and to reciprocate the transfer of the gene from species 2 into species 1 through these back hybridizations. And you can see in the two right-hand panels, two right-hand pictures, the effect of that transfer, the transfer of those genes. You can see that the flowers now have a very distinct difference in coloration. And that led to a change in pollination visitors. And the level of difference is huge, 68 to 78-fold change in visits in terms of birds to this one. Sorry, I've got that backwards. So birds to this one. And so this was a 68-fold increase by birds, and it was a 78-fold increase by insects. I think I've got that around the right way. I may have it backwards. This one, always trying to remember which way things go in this one is always a little bit of a challenge. And I did look this up before I began the presentation, and I think I've got it the right way around. If I've got it the wrong way around, please pardon that. Double check me in the book and make sure that I've got this the right way around. But remember, the deal here is it's a single gene change. A single gene change can change the nature of the interaction of these flowers with their pollinators. And that could be all that's necessary to start a divergence event that could lead to speciation. It doesn't have to be anything huge. It doesn't have to be a major change. It could be a single mutation like the snails. It could be a single gene like these flowers. It doesn't take much to drive a divergence. And if we want to add in on top of that the work of the Grants, I've mentioned Peter and Rosemary Grant to you before, and their observation on the island that they've been working on of the emergence of a new species of finch over the period of the last 15 years, simply because of the arrival of a single male bird from another island. And that male bird happened to find females in one of the two species that he could mate with and have offspring with. And those offspring could mate with themselves, could mate with their father, could mate with their mothers, but could not mate with, well, actually they could mate with each other. I'm not sure how much they could mate outside, but they've become reproductively isolated from the other two species of finch on the island, and they now represent a new species of finch, a third species of finch that has not existed before. Big Boy, the male who came in, has, by his very presence on the new island, created a divergence event that has led to the emergence of a new species. And as I said, I'll put the links to the videos. They're in the study area of Mastering Biology. I'll try and create links to those videos in the resources section for this chapter so that you can go and take a look at them if you have not already. I would strongly recommend that you go and take a look at them because it's a remarkable piece of work, and they go into detail about their observations of Big Boy and his partners and his offspring and how they have given rise to this new species of finch that they're documenting on the island. So one more concept check and then we're done for the day. So speciation can occur rapidly between populations once they begin to diverge from each other, but the time between speciation events can be much greater. How can this apparent contradiction be explained? This is what I was talking about earlier. Remember that speciation does occur quite rapidly once divergence begins, but there could be great lengths of time between those divergence events. And remember that the time between speciation events is not just the distance between emergence events. It's the distance between the beginning of one divergence event and the beginning of the next divergence event. So it includes that short period of time during which divergence is rapidly giving rise to a new species. So what's a good way of diagramming this, perhaps? Let's see if I can do this. So if this is the timeline, let's see if we can make this work. It's the first time I've drawn this, so you may have to bear with me for a minute. So speciation begins here. And this is the next speciation event. Change the color. So hopefully you understand this diagram. The period of divergence, the period during which speciation is actually occurring, which is the period of divergence, is a subset of the time interval between speciation events. So the time period between speciation events could be a long period of time, could be millions of years. But the period of divergence as a subset of that time is a relatively short part of that time, because divergence occurs very rapidly compared to the time it takes for new divergence events to begin. This is also part of the Eldridge Gould punctuated equilibrium model, where you've got a short period of time that represents the emergence of the species, and then a long period of time of equilibrium where the species remains relatively unchanged. The punctuated period is that period of divergence on this timeline. The equilibrium period is, let me see if I can diagram that in. It's this one. Yeah, I've got it. So this is equilibrium. The period during which the species remains unchanged. And then this is going to look awful messy. I'm sorry about that, but I'm going to put another one in. So hopefully that diagram makes sense in terms of what the question is asking, and helps you understand a little bit more about what is happening in terms of the emergence of new species over geologic time as we observe it in the fossil record. Anyway, that's it for now. That's the end of this chapter. As always, if you have any questions, concerns, or comments about anything that I've covered in this presentation, by all means, shoot me an email or come to office hours, and let's get any misunderstandings cleared up. Otherwise, I'll see you back here again for the next presentation when we start talking about chapter 25. Until then, bye for now.